Hello dear students. So today we are going to discuss the topic oogenesis of the chapter human reproduction. What is oogenesis? It is the process of formation of mature female gamete or egg formation. So where this process of mature female gamete formation occurs? It occurs in ovary. तो ये है ovary. Ovary, if you see the structure of ovary, you have an outer layer which is called as cover, and inner layer, the inner part, the main body is called as stroma. Stroma है. Stroma has this outer part which is dense called as cortex, and then inner part which is less dense, this is called as medulla. So if you see a section of ovary, then we see that there are many developing structures, and here this process of oogenesis takes place. Oogenesis यहाँ पर हो रहा है. If you see the cover, the outer layer, it has three layer. The outer layer जो बाहर का layer है, this is called as visceral peritoneum. Visceral peritoneum. And then inner to this we have germinal epithelium, and the innermost layer is called as tunica albuginea. तो इसमें तीन लेयर हैं. Visceral peritoneum has flat cells, germinal epithelium has cuboidal cells, and these are actually the germ cells which will finally make the female gamete later on. And tunica albuginea is a layer of connective tissue. It is a fibrous layer. So these germ cells, these germinal cells in the fetal stage, they divide and make lot of oogonia. So these cells are cuboidal here in the layer, but later on they make lot of oogonia through the mitosis in the fetal stage. Now let me tell you one more thing. This process of oogenesis has three parts. There are three stages or steps of the oogenesis. First stage is multiplication stage, multiplication or the mitosis. Here the cell divides and makes lot of oogonia. The second stage is the growth phase, and third stage is cell division or maturation division. Here actually we are talking about the meiosis, which finally makes the haploid egg cell. So the first part, multiplication, occurs in the fetal stage of the ovary. Then growth and cell division or meiosis, they occur simultaneously later on. That we will be discussing subsequently. हम लोग इसको डिस्कस करेंगे. तो अभी थोड़ा सा समझते हैं कि fetal stage का मतलब क्या है. Suppose there is a woman, her name is Geeta, and she marries Gagan. They have several children. Suppose there is a girl, Rinki, there is a boy, Rinku, and Geeta, she has her mother and father. Her mother is, suppose, Sangeeta, and her father is Shyam. So now here we are studying the process of oogenesis for Geeta. गीता की ओवरी में जो एग बनेगा उसको हम समझना चाह रहे हैं. So when Geeta was inside the womb of her mother Sangeeta, that was the fetal stage of Geeta, and she started preparing her oogonia when she was in the womb of her mother, and this oogonia formed will make the egg which will fertilize with the sperm, and these two will make the zygote which will make their children, Rinki, Rinku or others. तो इस तरीके से ये एक मल्टी जनरेशनल थिंग है. द प्रोसेस ऑफ oogenesis starts right in the fetal stage and then it continues for several years. So now I hope that you have understood this. आगे बढ़ते हैं. These oogonia, because they divide by mitosis, they are diploid. Initially, during the first month of at the end of the first month of fetal life, there will be around 1700 oogonia which are formed, and by the end of third month, there are 7 million oogonia formed in the ovaries. 70 lakh. 3 महीने के फिटस में 70 लाख oogonia. And then these oogonia start degenerating or dying. This process is called as atresia. And by the time of birth, it has only 2 million oogonia. And during the fetal stage itself, this oogonia, they start getting surrounded by a layer. This layer is called as granulosa cells. Initially they are flat, later on they become cuboid. When they are flat, they are also called as pre-granulosa cells. And this follicle in the fetal stage then further grows and then it is called as primary follicle. तो प्राइमरी फॉलिकल में क्या होता है? एट द टाइम ऑफ बर्थ एक गर्ल के अंदर अराउंड 2 मिलियन ओवरीज, 20 लाख ओवरीज होती है. व्हाट इज द कैरेक्टरिस्टिक ऑफ प्राइमरी फॉलिकल? हियर the oogonia has started the growth and it has started the cell division or meiosis. In the fetal stage itself, the growth and meiosis starts, but this is arrested, it is stopped at the prophase 1. Prophase 1 of the meiosis, and this grows in size, and now this cell, this oogonia which has started the cell division is called as primary oocyte. So primary oocyte is still diploid, surrounded by a single layer of granulosa cells. Single layer of granulosa cells. तो ग्रेनुलोसा सेल का फंक्शन क्या है? व्हाट इज द फंक्शन ऑफ ग्रेनुलोसा सेल्स? दे आर प्रोटेक्टिव इन नेचर, दे प्रोवाइड नॉरिशमेंट और सपोर्ट. दिस इज द मेन फंक्शन टू द डेवलपिंग oocyte obviously, and they also secrete estrogen. So these are the functions of granulosa cells. Primary follicle is characterized by single layer of granulosa cells. It also has an extracellular layer made of lipoprotein called as zona pellucida. Now the primary follicle further grows, and this further growth and development of the follicle actually starts only at the puberty. तो हमने देखा कि oogonia का फॉर्मेशन तो फिटल स्टेज में हो गया था, 20 लाख oogonia बन गए एट द टाइम ऑफ बर्थ, बट इनका फर्दर डेवलपमेंट जो होता है वो स्टार्ट होता है ओवरी में ओनली कब? जब प्यूबर्टी आ जाती है, दैट मीन्स अराउंड 11-13 ईयर्स ऑफ एज. तो एट द एज ऑफ प्यूबर्टी these follicles start development, the follicular development and maturation again starts. So if you see a section of ovary, you will see that there are many primary follicles which are developing, then there are many even larger ones which are called as secondary follicles, then many are even larger which are called as tertiary follicles, and then few mature follicles are also there, and finally in every month, during the every menstrual cycle, at the end of at the middle of the every menstrual cycle, you will have only one single mature egg which will be formed. Okay. So now let us study that how during the menstrual cycle or during this follicular development, how this follicles further develop. The primary follicle has primary oocyte which further grows, and then the granulosa cells now become several layered, multi-layered granulosa cell, and another layer called as theca. This theca is also developed, and this becomes multi-layered granulosa cells, and this stage is called as secondary oocyte. तो सेकेंडरी oocyte में क्या होता है? सेकेंडरी फॉलिकल में दो फीचर हैं, ग्रैनुलोसा सेल्स के सेवरल लेयर्स और एक एक्सटर्नल कवर कनेक्टिव टिश्यू, कुछ सेल्स भी इनसे बना हुआ लेयर थीका ये बन जाता है. दिस फर्दर ग्रोज एंड डेवलप्स एंड इट फॉर्म्स टर्शरी फॉलिकल. Now, what is the feature of tertiary follicle? In the tertiary follicle, this primary oocyte, this further grows in size, and then this theca, it further differentiates and it makes theca externa and theca interna. दो लेयर बनाता है, and this is the primary oocyte. One more important thing here, a crack or an space develops between the granulosa cells, and it starts making a cavity. This cavity is called as antrum, and antrum has a fluid which fills this cavity, and this fluid is called as follicular fluid or liquor folliculi. Liquor folliculi or follicular fluid. So what are the characteristics of tertiary follicle? Tertiary follicle has a primary oocyte, it has a zona pellucida layer, it has an antrum, and theca has now differentiated into theca externa and interna. Tertiary follicle further develops and makes now the mature follicle. This mature follicle is also called as Graafian follicle after a name of a scientist. Graafian follicle. If you see a section of ovary, you will see many Graafian follicles. तो Graafian follicle की प्रॉपर्टीज क्या है उसको देखते हैं. Here, now the primary oocyte which was growing, it was arrested at prophase 1, it completes the cell division, and now it forms the secondary oocyte. The secondary oocyte is haploid in nature. So we can say that in the primary follicle, in the secondary follicle, and in the tertiary follicle, we have the diploid cell, the diploid primary oocyte. But in the mature follicle, we have the haploid secondary oocyte. It has now completed the meiosis 1. And again it has started the meiosis 2. But this further gets, meiosis 2 starts, but it gets arrested at metaphase 2. So again it is stopped at metaphase 2, and this cell division is unequal cell division. The majority of the cytoplasm is retained by the secondary oocyte, and only a scant, a little bit of the cytoplasm is given to this another second cell which is called as first polar body. This is also called as polocyte. This is also haploid. Later on this degenerates. And the antrum, the cavity further grows and lot of liquor folliculi is there. We can notice one more point about this antrum, that it is not centrally located. इसका जो पोजीशन है एंट्रम का, इसको एक्सेंट्रिक पोजीशन बोलते हैं. एक्सेंट्रिक, it is eccentrically located, it is not located in the center. So what are the features, what are the properties of a mature Graafian follicle? Here the primary oocyte has completed the meiosis 1, so it contains the secondary oocyte. It has the theca externa and theca interna. The antrum has grown quite large with lot of liquor folliculi, and there is one more structure here, this one. These granulosa cells which are present between the antrum and the primary or the secondary oocyte, this structure is called as, the solid mass of granulosa cells is called as cumulus oophorus. Cumulus oophorus कहते हैं इसको, mass of granulosa cells को. So now these mature follicles, they are ready to go to the next stage. At the mid of the menstrual cycle, that is around the 13th or 14th day, there is a sudden increase in the level of luteinizing hormone. This is called as LH surge. LH surge बोलते हैं. और इसकी वजह से क्या होता है, this mature follicle now ruptures. This rupturing is called as luteinization. So LH surge leads to luteinization of the mature follicle, which results in the release of secondary oocyte, also called as ovum. So this process is called as ovulation. So when the ovulation occurs, what happens, that this secondary oocyte which is covered with the zona pellucida glycoprotein layer, and these granulosa cells, ये granulosa cells का जो layer है, this is released. So now there is a characteristic shape of this released secondary ovum which also has the granulosa cells. इसके जो लास्ट के पेरिफेरल सेल्स होते हैं, ये थोड़ा सा रेस की तरह से बनाते हैं. That is why they are, this structure is called as corona radiata. So this structure is called as corona radiata. Okay, and this is the released ovum. Now mind here that it still has not completed the meiosis 2. So once this secondary oocyte, or NCERT also calls it ovum, this is released, you know that just near the ovary there is this finger-like structure called as fimbriae in the infundibulum. तो वो fimbriae उसको इस तरह से अपनी तरफ खींच लेती हैं. Then this oocyte or egg, this enters into the fallopian tube, it travels further, then it reaches the junction of isthmus and ampulla in the fallopian tube, and there when the sperm comes and attaches with the zona pellucida layer. Now what is the function of zona pellucida layer? Let us see here. Zona pellucida has many functions. NCERT says that tertiary follicle has this characteristic zona pellucida layer. Okay, zona pellucida is made up of glycoprotein. So what is the function? First is, it is protective function. It protects the oocyte which is developing. Then it prevents the polyspermy. Polyspermy means it will not allow several sperms to fuse with this ovum. It will only allow a single sperm to fuse. And then it also ensures the species specific fertilization. That means if the sperm belongs to some other species, not this species, then this zona pellucida will have certain chemicals, and these reactions will prevent the entry of the sperm of other species, and it will ensure that only its own species makes the successful fertilization. So once the sperm has arrived, upon arrival of the sperm, when it reaches the ovum, then again there are certain reactions which are triggered. Upon arrival of the sperm, there are few things. There is one factor called as M phase promoting factor. This is broken down upon arrival of the sperm. And there is another factor called as APC. This APC is called as anaphase promoting complex. APC का मतलब anaphase promoting complex. MPF का मतलब M phase promoting factor. So upon arrival of sperm, MPF is broken down and APC is activated. It is activated. This leads now the exit from metaphase 2, and it now resumes the meiosis 2 and completes the meiosis 2 to make the second division of the meiosis 2, which now will result into the formation of ootid. So from the secondary oocyte, after the second meiosis, now the ootid or the real ovum will be formed. And again there is the second polar body will also be formed. This second polar body again results because of the unequal cell division. The majority of the cytoplasm will be retained by the ootid. These are very small bodies. The first polar body and second polar body, they will later on degenerate. In some animals, the first polar body also continues the division, and along with the secondary oocyte, the first polar body also divides and it makes two polar bodies, so there are total 3 polar bodies. NCERT says that we are not sure, we are still don't know that what happens in human, but in most of the textbooks you will find that the first polar body in humans does not divide further. So humans have only the first polar body and second polar body, which later on degenerates. Now this ootid, which has just immediately formed only just upon the arrival of the sperm has the nucleus which has just entered here. It undergoes the fusion and fertilization immediately to form a zygote in the fallopian tube. This zygote still has the zona pellucida layer. Zygote now starts cleavage divisions, and then from one single cell, several cells are made. First the morula stage, then blastula stage, and so on, that we will study in the development of the embryo. But the zona pellucida layer, this persists till blastula stage. It is the blastula stage at which the implantation occurs. That means ये फर्टिलाइजेशन के बाद फैलोपियन ट्यूब से मूव करते हुए यूट्रस में जाएगा, उसके एंडोमेट्रियम में ये ब्लास्टुला स्टेज पे जब पहुंचेगा तब इम्प्लांटेशन हो जाएगा. एंड देन दिस in the blastula stage the zona pellucida layer starts rupturing. Okay, so this is the entire process of oogenesis. Now let us again revisit. We had said in the starting में हमने बोला था कि there are 3 steps or 3 stages of the oogenesis. पहला क्या था? Multiplication, which occurs in the fetal stage. Lot of oogonia are formed by mitosis. This is the multiplication stage. Second stage was growth, and third was the maturation divisions or meiosis. So you can see here that the oogonia further grows and it starts the cell division. It resumes the cell division which has started in the fetal stage and it is finally completed in the fallopian tube. So it takes several years. Okay. One more thing here, I would like you to notice and remember that. See, this is the primary follicle, secondary follicle, and tertiary follicle. All these three follicles, they contain primary oocyte, and primary oocyte is diploid in nature. The mature follicle or Graafian follicle contains the secondary oocyte, which is haploid in nature, but still it has not completed the meiosis. When the ovulation occurs, the secondary oocyte is released, though it is called as ovum or egg, but still it is not the real egg, because it has not completed the meiosis. The meiosis completed only upon arrival of the sperm. So this is another point. You also can notice that there are these two stages where this antrum is developed. So these follicles which contain the antrum are called as antral follicles. Okay. So in the lifetime of a woman, probably we can make a rough calculation that if we suppose that there is at 13 years of age a woman starts its puberty, and if we say that she continues to produce up to about 50 years, if she continues to do the menstruation, we can say that roughly up to the 40 years she keeps on undergoing the menstruation every month, and every month the ovum will be released. Obviously, when there is a pregnancy, this cycle stops, and then each month it will not release the ovum. So in a reproductive lifetime of a female, that is in around 40 years, a single woman produces around 12 into 40, that means 480. So we can say that in a lifetime of reproductive stage, in a reproductive lifetime rather, a woman produces about 500 mature ovum or egg. Okay. So at the time of birth, 20 lakh, 2 million primary follicles were there, which further keeps on degenerating. At the time of puberty, only she has 60 to 80,000 primary follicles at the time of puberty, and out of those 60 to 80,000 primary follicles in each ovary, only about 500 are utilized to make the mature egg. Okay, so now we will stop here. Thank you.